Tracking early onset Alzheimer's. Before I start, I want to welcome you and thank you for coming to this presentation. So who are we? Hamline Biotech is a rising biotech firm located in Como Park in the Bandana Square complex. We have 5 years of experience and 123 staff members. We have 15 initiatives in process, 8 preclinical trials, 4 phase 1 clinic trials, 2 phase 2 clinic trials, and 1 phase 3 clinic trials. FDA approval received in early 2019 for a new gene therapy, which replaces the mutant BRCA gene with BRACZ, is now rolling out to doctors and potential patients. You may have seen the TV commercials and advertisements along with our viral YouTube video and animated presentation. This is our current team. Oliviana Cervantes is our team leader, owner, and CEO of Hamline Biotech. They have a PhD in biology from Boston University, along with a Master of Art Therapy from the School of the Art Institute of Chicago. Next, we have Rosie Larson, our clinical lead, Director of Clinical Research at the Hamline Biotech. Worked as a clinical research scientist at McAllister Biotech for 3 years before she joined us at Biotech when we opened 5 years ago. She has a PhD in biology from the University of Minnesota. And finally, we have Felix Sanchez, our administrative lead, MBA from St. Thomas, worked at Augsburg Biotech for 5 years before joining us at Hamline Biotech. So what's the big idea? According to Alzheimer's News Today, an estimated 200,000 people in the United States have early onset Alzheimer's disease. Early onset Alzheimer's usually appear in people between the ages of 30 and 60 years old, and the typical life expectancy after an Alzheimer's diagnosis is only 4 to 8 years. We estimate 33 new diagnoses of early onset Alzheimer's per year. That is 200,000 divided by a median of 6 years. According to the Dana-Farber Cancer Institute, there are 3 gene mutations known to cause early onset Alzheimer's by producing a toxic protein fragment called amyloid beta peptide, which builds clumpy plaque in the brain, which results in Alzheimer's. These are the gene mutations that cause early onset Alzheimer's, and we have created 3 new genes to replace these mutations. The genes that we have created are APPZ, PSEN1Z, and PSEN2Z. So how would this work in the real world? Assuming that we have FDA approval, the steps would be as followed. Potential clients would take a gene test such as 23andMe based on family history or simply curiosity. Clients learn that they have at least 1 gene mutation and goes into a physician. A physician offers options of gene therapy and identifies risks and the cost. Client undergoes such gene therapy where a vector is introduced with a virus such as cold virus along with the replacement genes. Replacement genes start to take effect and the client retakes the test and the mutated gene does not show up anymore, and thus the celebration commences. So what is our plan? According to biotechforecast.com, clinical trials and FDA approval are a very long process and take years. So here's a quick summary of the process using our new ideas as the example. Step 1. Preclinical trials on lab animals can be done in-house at our facility in Como Park. A plan has been developed. Step 2. Document results and request FDA approval for clinical studies on humans along with Institutional Review Board (IRB) approval. If approved, IRB will provide guidelines for the trials. Step 3. Assemble panel of physicians and researchers to supervise clinical trials. Early discussions are occurring to bring in a stellar panel. Step 4. Phase 1 studies, safety. 80 screened volunteers test the gene therapy to review for human consumption along with side effects. Document and proceed if safe. Can be done in our Como Park facility and meet with the FDA as needed. Step 5. Phase 2 studies, effectiveness and safety. 200 screened volunteers test the gene therapy for human consumption. Side effects and effectiveness. Document and proceed if safe and effective. Can be done in our Como Park facility and we must meet with the FDA as needed. Step 6. Phase 3 studies. First, meet with the FDA to discuss prior results. Then screen 1,000 volunteers for safety and effectiveness. We need to contract with a larger facility. Document and proceed if safe and effective. Step 7. Biological license application to FDA with all results and an outline of manufacturing and marketing safety. If file approved, then biologics evaluation and research reviews results and label for approval. So, what are the challenges that we're going to combat? Striving for testing protocols that include sufficient reviews for 3 mutant genes and include information of the new gene therapy. Strive for health insurance coverage of this new gene therapy. Strive for timely manufacturing to make product available as needed and avoid any issues with the gene expiration. And strive for marketing that reaches 33,000 potential clients per year and their physicians that explains this new life-changing therapy. Next, we're going to talk about funding. The National Institute of Health has agreed to fund Phase 3 and FDA approvals after Phase 3. What we currently need help funding for is preclinical testing, Phase 1 testing, Phase 2 testing, FDA approvals prior to Phase 3, marketing, and manufacturing. We are willing to offer market shares for any funding assistance, and if you are willing to fund, please have any proposals submitted by July 30th, 2019. Thank you so much, and we hope to see you in the future.